gonna record. Okay.
Okay.
So I'm gonna actually start by just introducing myself a little bit. So I'm Louis. So I was actually born here. So that's in the south of England. And that's Southampton, which is a port town. And I think the most famous ship to have sailed out of Southampton is the Titanic. So I illustrated there with the Titanic.
Yes, yes.
But then, I grew up in Lisbon. And so that because my family is from Portugal, and some of people might recognize from my name, that's a Portuguese name. And then that's where I went to undergraduate and master's, and I studied actually computer science. So this was pure computer science, had nothing to do with biology. But at the time I also started to attend these biology seminars, and I got a bit of biology FOMO because I thought that some of what the biologists, particularly the computational biologists, were doing was more interesting than what I was doing. So I decided that I wanted to do that instead of what I was doing. And so I went to Pittsburgh, Carnegie Mellon, to do a PhD, and that was a PhD in computational biology, where I did about half the time in the lab, so wet lab, growing cells, doing experiments, and half the time developing computational methods and writing code. So after that, I went to the EMBL in Germany, and that's actually where I switched from working in this field that I was working on before, which is eukaryotic cell biology, and I switched to working in the microbiome field, which is what I do nowadays. So I spent 5 years there. Then I kept going east. I spent 5 years there in Shanghai, where I started my own research group at Fudan University, and then last year, so that was actually almost exactly a year ago, it was in September 2023, I moved my research group to QET, so in Brisbane. So that's where I am now. I'm actually, now it's a holiday period. I'm actually a little bit further south in a place called Coffs Harbour. But this is my journey a little bit also. I started as a computer science, then went into the lab for my PhD, did a lot of lab work, but then since then, most of my work is purely computational, but it's now more biologically applied.
Thank you, Prof.
So
maybe a little bit about
please.
that. The group here is also multidisciplinary. We have computer scientists, we have medics, doctors, we have people from statistics, mathematics. Yeah, so that's the background.
Yes. Okay, that's great. I mean, so in my own research group, I mean, I also try to have people who come in with very different backgrounds. I think that's so. For example, here in this slide that I'm showing, so Svetlana. So when she joined, she was coming. Oh, sorry, I'm just highlighting her. So when she joined, she was coming purely from a wet lab biology background. So she knew how to grow bacteria, etc., but she had never done any programming, and and then she joined, and now she's not a computer scientist, but she can analyze data and do that type of work. And Vedant is a little bit coming from the other side. So Vedant was, so he's very junior, very young. He's now going off to college this year. He's coming from this computer science software development background. Right. So actually, I'm just going to present a couple of different projects that we do very briefly, and I'm happy to talk more about them. But otherwise, I will just present them very briefly, and then there's 2 projects that I'm going to present, including the one that motivated this talk, this exploration of the small proteins. Those I'm going to present in depth, but I'm going to just present a couple of little things that I'm happy to talk about. So I just wanted to have them, and if people want to ask questions afterwards, we can discuss them, or we can discuss. But otherwise, just so one of them is antibiotic resistance project, which we've done. This is a big collaboration. Actually, I should have updated this, because since April we also have a group in Nigeria, where we work with people from around the world, and the big thing that we've been looking at is, so there's many tools to do this, actually. There's many tools that were developed, but so there's two issues. So the first one is already that the tools use very different nomenclatures and names, and this includes things that would be, it just would be very easy for a human to understand that, for example, that a tool might spell it van-r, and the other tool might spell it van, -r, but sometimes the names might be completely different, in which case you would only know it's the same if you have some sort of a lot of background knowledge. And so our solution here is to use an ontology, and we didn't come up with this ontology, but we developed a tool that basically maps everything to the same ontology. And this is a tool we've called argnorm. And part of this is because we've been looking at comparing these tools, and so in this case, this is a very comparison of different habitats. So here, and again, a lot of the data I'm going to be showing you, this is metagenomics data. So this means that we or someone went out and collected a sample, for example, from the marine environment. What you do is you take, it's a bit fancier than this, but you can imagine that you take a bucket of water, and it's just a very sterile bucket, but you then filter that water through a filter, and then so that the water goes through and the bacteria get stuck on the filter. And then you have that sample of all of the bacteria that were present in that 20 liters of water that you filtered, and then you can sequence and you can ask what's there, and you can ask how many different antibiotic resistance genes do you see in this community. And so here we have 3 different ways of doing this computationally. So this is the same data, it's just a computation analysis that changes using a database card with a tool called Abricate, or the same database with a different tool called RGI, or a completely different tool called Deep ARG. So all of these are very widely used, and you can see that not only do there's some variation that's to be expected, but even the relative standings of the different habitats change. So if I use on the very left, I would say, there's almost nothing in the marine environment, and in freshwater there's a little bit more, but it's wastewater and human gut. But if I use the second tool, then I would say, actually, no, there's a lot in the marine environment. It's worse than freshwater, but still not as bad as wastewater or the human gut. And if I looked at the far right with Deep ARG, I would say that actually wastewater is much worse, but the human gut and the marine environments are kind of equivalent. And so here it's not. So we just wanted to make the point that these different tools, even though often in manuscripts and papers people don't, these details are somewhere in the methods section, but these very technical details, they can completely change your message and your results. So another project we have, and this has been led by Anna, is we've been looking at dog gut microbiomes, and here we're using these 2 sequencing technologies, Illumina and Nanopore, and we are analyzing this. But basically our message right now is that we've gotten this very, very good reference for at least pet dogs, both in Shanghai, and we've analyzed that they seem to cover very well dogs also from other parts of the world. We've done a similar sampling study of public parks. Here, this was also in connection with antibiotic resistance. So the goal was to try to understand what the public parks are. It's an environmental sample, but there's also obviously a lot of human contact because people are you can see people are having picnics, kids are playing. And so we are people are constantly being exposed to the organisms that are there. And so we wanted to ask, is there something really dangerous in these public parks? And we looked in Shanghai, and then we also had collaborators from Nanjing who contributed samples from that town. So Shanghai and Nanjing are about 300 kilometers apart from each other in China. They're both very developed cities in the richer parts of China, but they are 300 kilometers apart. And finally, these rapid fire projects, we've been looking with Christopher Salen, who's from Stockholm University. We've been looking at this tool called Strobaline, which we were very, very impressed with when it came out. It came out about 2 years ago, but it didn't really work for metagenomics. For some things, it didn't really scale very well, and so we've been improving it and adapting it for metagenomics, and we actually have really good results. And we have the code. Everything's available. We're just writing up a manuscript, but people can use this code already. All right, so these were just some rapid fire. Oops, sorry, but some rapid fire projects that we have. But now I want to go into these. I want to present basically 2 projects that we've been developing. So one is this vision of trying to look at the global microbiome as a whole. And so this actually goes back. So here, this was published in 2022, but this actually goes back several years, where decided to to try to look at the microbiome, not just take this sample here and look at this environment there, but try to look at everything that was available and try to put everything together and try to see if this could help us make sense of things. And so we say we want this global microbiome perspective, in the sense that there's not a single, everything's a little bit connected. So at the time, this was everything that we could find in the public databases. So a lot of our work is built on looking at what's available in public databases, and we found just over 13,000 metagenomes from 14 habitats that we thought were good enough. And here, good enough mostly means technical metrics of quality. So this was what we were looking at, is from a technical perspective, these were the ones that we thought were good. And then we processed them, and we processed them consistently. So every single sample went through the exact same pipeline. And the first thing we did was we built these contigs and extracted open reading frames. So genes. And because we are going to obviously have the same organism living in multiple locations, that means that we are going to have the same sequence present in multiple locations. So we dereplicated that. So here, dereplicated means we had the exact same sequence. So now we had 1 billion of these dereplicated open reading frames. But then we wanted to look at them at a little bit of a higher level. And so we clustered them, allowing for some divergence. And we did this, and there's a couple of different levels, but the main ones are this 300 million species level unigenes. So this is a 95% nucleotide clustering, which for bacteria more or less corresponds to a species. So humans, we are almost all the same. So right, we have around this room, or Zoom room, right? We are all basically 99.9% identical. But bacteria, there's a lot more divergence, and so in a bacterium, 95% identity is still the same species. And then at the other extreme, we cluster them so that we put two sequences together if there's any statistical evidence that they are homologous. So if there's any statistical evidence that we can claim that they have a common ancestor. And so there we call these protein families. So some of these, they can be very divergent, but they somehow still, maybe billions of years ago, they have the same common ancestor. So here, just try to justify these thresholds that we use. So this 95%, again, it's trying to replicate a species threshold, and where we've benchmarked this by looking at, so if we have the same sequence within the same species, or the most similar sequence within a genus, so that's the next level up, but not the same species, And there's a bit of overlap. There's no, so all of these thresholds, nothing's going to be perfect, but 95%, which is a very traditional one, it does seem to kind of hit this mark very well in, in separating where these two lines meet. So the first thing we have here is we have this resource. So we have these 300 million sequences, and we've built these mags. Actually, I'll talk a little bit about mags, and so this was a resource that we've made available, and I think and others are also using this to investigate the global microbiome. So, but we wanted ourselves to also explore it a little bit. So and you'll see that this is a little bit our modus operandi, which is we build these resources, and then we run our own analysis. But we often also realize that this can be very valuable for others, and so we also make them available, and we hope that people are also taking advantage of them as resources. So the first question was, okay, we put it all together because the hypothesis was that there's some links between these things, but we wanted to actually check and quantify this. And so here in this graphical representation, so the width of these ribbons shows how much sharing there is between different habitats. And so on the left, you can see these are the major groups. So between, say, the mammalian guts and other mammalian habitats, between environmental environments, so marine, soil, a little bit more with the built environment. And so we can see that actually there is most of the share. So most of the genes are specific to a single one of these groups. Although there is sharing, but if we zoom in here on the right side, and let me just zoom in. So this is so if we now just look within the mammalian guts or human habitats, we can see that now there's a lot more sharing. And so in particular, the dog gut, we had already back in 2018 compared then this comparison where we had argued, or we had seen that the dog gut shared a lot with the human gut, but now in this updated study, this is still true, but we can see that actually the cat gut shares even more. So it seems that at least within these animals that we're looking at, it's cats and dogs share the most with humans. It's not mice. Pigs a little bit more than mice, but it's actually our pets. And so our thinking is that this is mostly because of lifestyle and the fact that these animals have now for generations lived in permanent contact with humans, and so that there's been some convergence of between their microbiome and ours, and there's also some convergence between, or some sharing between different body sites, and so the message is that on the one hand there is most of the genes are habitat specific, so we will find them in a single gene in a single habitat, but if we look at more similar-ish habitats, so for example different mammalian guts, then we start to see sharing. And unfortunately too, if we look at what kind of genes are shared, it was not surprising, but we confirmed that in fact things like antibiotic resistance are more likely to be shared than other genes. We also found this, and I keep showing this. For me, it's a curiosity that I don't think we fully understand the mechanisms here. As I said, a lot of what we do is we're trying to look at these explorations, but we don't. But we don't solve everything ourselves, and I'm putting it. Maybe someone else will solve this. So again, we're collecting these samples, right? And in a sample, we are going to find some organisms, and they will have, and we will detect a certain number of genes. Sorry, and the more genes we detect, Actually, give me a second. I just need a glass of water. I'll be right back. I'm really sorry for this.
Sure, sure. It's okay.
Yeah, all right. I'm sorry. I'm back. Sorry. I was starting to cough because of dry throat. So, all right. So what we were doing here. So in a sample, we can count how many different genes we see and how many different species we see. And these are two different metrics of diversity. And then we can do this, and we can do this ratio between the two. So, and on the right side, we can also see, how many different unit genes we have compared to how many protein families we have, and we can do the ratio between these two. And the point, especially here on the right, on the left, is that we see these peaks, right? And for example, the human gut. So the human gut, this includes samples from many different studies from different parts of the world. From people who are younger, people who are older, people who are healthy, but still there's a relatively well-defined peak that is a different peak from the peak of cats. And it seems that and this seems to be the story for all of these habitats, except maybe the marine and the soil environment. So in the marine environment, maybe you could see, okay, there's some sort of modality there. And in the soil environment, it seems that, maybe there's just spread out. But even actually, this is very misleading because what happens is that we were calling marine a bunch of different things. And if I split up the marine environment into the surface waters and this deep chlorophyll maximum, which is a little bit deeper than the surface, but there's still light there, then this you get one peak, and then we have this mesopelagic layer, which is a little bit deeper, where now it's dark. Then we have a different peak there. And then in the soil environment, so this is not my area of expertise, so what the people who know more about soil have told me is that these are just different types of soil, and they have different pH. And so I think here the problem is that we have all of these different things, and we say, oh, this is soil, but in fact we should be saying this is this type of soil and this is that type of soil, and they will have very different. This will give rise to very, very different. This will give rise to very different communities, right? And so this is what we're seeing. So this is one of these things where I'm still not sure exactly what are the mechanisms that are generating this observation, but we did see this pattern that just these basic metrics of diversity do seem to separate the habitats quite well. Right, so this was published back in 2022, and then we were asking ourselves, so okay, so where do we go from here? What do we do next? And on the one hand, we wanted to go bigger, so we wanted to look at more data, and in particular, in the meanwhile, we say we're looking at the global microbiome, but obviously we're always limited by what others have sequenced. We do a little bit of sequencing ourselves, but it's a tiny amount compared to everything that's been done before. So we took another snapshot of the data, and you can see, first of all, we have a lot more dots.
Okay, there was a connection break there, but I think you're back.
I think I'm back. Yes, I'm sorry. And also, you could in the first map I showed you, there were certain parts of the world were completely not represented, including the continents of South America and Africa. And now I think they're still not represented proportionally to ideally they'll be represented proportional to their population. but at least they are starting to be more represented, and so we're starting to get sampling from more spread out around the world. And also we have more and more different types of habitats. So here I've grouped them to be able to just plot them, but there's actually 75 different categories now. This includes a lot more animal microbiomes as well. And we've published this just earlier this year in a database. So this database is called SPIRE. So SPIRE being basically the public version of all of this data, including the metagenome assembled genomes and all of these annotations. But the other thing we wanted to do was to look smaller and to look at small proteins or microproteins, because everything I was showing you before, if we never really talk about it too explicitly, but we put it in the methods. We're not, but we don't make it. Is that the small proteins we kind of ignored, and so this comes at different stages in the pipeline, and we did that, and actually almost the whole field did that, basically with the exception of Ami Bhatt's group in this paper from 2019, which was the only at the time was the only only effort looking at microproteins at the large scale. And the problem with small genes is that everything is just harder. And the statistics just become very difficult. You just don't have a lot of observations there to the number of amino acids, and so for us we're just saying that below 100 amino acids, that's small proteins, and often just the statistics don't work so well. For example, let's say if you have some random DNA, and you have some area where you have some start codon and then a lot of hundreds of amino acids and then a stop codon, that's not going to happen by chance, because mutations are going to be introducing stop codons in the middle, and if you don't have evolution to remove those from the population, then this will not stick, right? So this will get purged from the species. So if you see a big stretch of DNA without any stop codons, at least there's some conservation signal happening there. But this logic breaks apart if you have a short sequence. I mean, you could just by random chance you can have a short sequence that's looks like a protein, but is very short. That could literally just be random chance. So a lot of our techniques that we've developed as a community of bioinformatics for the last 30 years simply do not work very well anymore. And so with Celio and Ichian, we've been looking at this at least, and Ichian's paper was just published a couple of weeks ago in Nature Communications describing this effort. Again, this is the same map we took before. So so there's, if you count very quickly, so there's 60,000 dots in the plot. And so each one of these again represents a metagenome, so a sample that was taken. We collated it with 90,000 microbial genomes, and we put everything together, and we had 4 billion of these small open reading frames. Again, we expect to sequence the same thing multiple times, so we collapse the redundancy, so we only keep one copy of each sequence, and then so here, and then that means that we have about 1 billion smORFs that are, so smORF stands for small open reading frames, that are identical at 100% amino acid identity, which then we can group into about 300 million at 90% amino acid identity. And so again, again, just having a look at what's there, so we can see that most of this is novel. So if you look at other databases, we can only find 5% of it that matches other databases. But on the other hand, if we ask the question the other way around, most of the other databases are contained in our catalog. So if we look at what's present in RefSeq for either bacteria or archaea, or in this smORFs is Amisbat's group's work, you can see that we cover most of what was there before. On the other hand, and I'm just going to be turning on the light as well. On the other hand, we can also do this study, this analysis where we look at the curves of, so as we sequence more and more, are we saturating? And the answer is no. Maybe in the human gut, because that's been more explored, we see a little bit that the curve is starting to a little bit saturate. But in the case of the soil or aquatic environments, it's still going up. So basically, any new sequence that we have, any new sample, is still contributing a lot of new small open reading frames that hadn't been seen before. Most of this, this is just, so we have this 100 amino acids as a hard threshold, as our definition of what it means to be small. And most of the catalog really is on the longer side, although we do have, again, we're looking at the billion sequences, so a small fraction is still a very large number. So we still have millions of sequences that are 20 to 40 amino acids. And a lot of what we're doing is, so we have this idea that if you have a sequence in one genome or in one metagenome, maybe that can be a false positive. But if we see it repeatedly in many metagenomes, then that's probably something real. At least, there's some conservation happening there, some biological conservation. And so we also make available this tool called GMSC-mapper that enables anyone who has a set of sequences to also try to find them in our catalog. So this can be run on the website, or you can download it and run it locally if you want. So again, in addition to wanting to make this available as a resource, we looked a little bit into trying to understand it ourselves, what's going on. And so one of the first things we looked at is what kind of organism is producing these small proteins. And obviously, organisms for which we have a lot of sequence, we will have more small proteins. And so we normalized this so that by what we're calling density. So this is how many small open reading frames do we see for each assembled megabase pair. And you can see that that there's some differences between different taxonomic groups. And here we've colored them by whether they're bacterial or archaeal groups. And maybe on the left it's not so apparent. So these are the top sort of densest groups. And it's about 50-50 between bacteria and archaea, which is actually quite extraordinary because most of the samples are bacterial. So on the right side now we split this up into bacteria versus archaea, and archaea simply have more small open reading frames in terms of density. But again, we only have this. So we have these sequences. We don't know what they do, and this is actually a big problem for the next several years that we'll be working on. But we wanted to start with one particular subcase, which is these antimicrobial peptides. So what are antimicrobial peptides? So an antimicrobial peptide is, as the name says, it's an antimicrobial in the sense that it's either outright kills or at least stops the growth of other bacteria. And it's a peptide, meaning that it's a short sequence, like a small protein. And so we were working, and again, Celio from my group, but then also Marcelo from University of Pennsylvania, were the people leading this effort. So we took all of the open reading frames that I showed you before, and we fed them to our tool called Macrel. So this is a machine learning based tool, and it classified them into a catalog of 860,000 AMPs that we're calling, sorry, the AMP sphere. And so this has also been published quite recently. Okay, I'm actually gonna skip this one. And and so this was all computational work. So we, and I can tell you, so we were working on this for several years. So this started in 2018. Doing all of this data processing, all of the analysis. We developed this tool, Macrel, which we had to develop because none of the existing tools really worked well for our use case. So we published that in 2020, and then we did all of this clustering and all of this crunching, and then we have these 1 million candidates, but so far we have actually this is all prediction. This is all, and we think we're doing a good job. We think we're trying to be conservative and cautious, but we don't really know. And so then we worked with Cesar Lafuente at UPenn and Marcelo, and so they tested our predictions, and this was sort of the moment of truth. And of 100 predictions that we tested, 79 came back positive. So we synthesized these molecules. We see whether they really are antimicrobial, and we tested them against both these very nasty pathogens and also other gut commensals. And then finally, some of them, the top candidates, we did this mouse model where it's a skin infection. So you apply the peptides kind of like as a cream, and here we used polymyxin B, which is a commercial antibiotic, which is also often applied, like as if you have a skin infection, this polymyxin B is applied. Actually, polymyxin is not used very much in other ways, but here, if you see, this is how much decrease there is compared to the initial, and we have at least these two. A few or several of them are very close to the positive control. So we have these preclinical models that seem to show that this could potentially be useful as an antibiotic. We also wanted to ask a couple more fundamental questions about the biology and the evolution. And so one of them is just, where do all of these sequences come from? And one theory that we had is that, so we know that there's sometimes AMPs are generated by the cell first produces a big molecule, a big protein, and then it leaves off a small part. And then we asked, maybe this could also happen at the genomic level, so that what we're seeing is a partial gene. And so what we looked for this is, we looked whether there was matches between our small proteins and full-length versions of proteins. And again, I'll caution that this is only 8% of, we could only find matches for 8%, but interestingly, about 1 in 4, the match was such that what we see is that there's a starting position that's shared, and then there's an early termination. And so here our model, and I'm just going to illustrate to this example. So in this example, so we have here, there's two sequences from, or several sequences from Prevotella genuni in green, and then Prevotella melanogenica in this purple, and then our peptide is this in metagenomic contig, and so they share this start position. And the sequences are almost identical. There's a couple of positions that differ, like the C and T, but then there's this mutation from a G to an A, which induces early termination. So now we have a truncated protein. And it just so happens, so our model of the world is that this happens quite often. You have these mutations. And in this case, it just so happens that that this truncation is actually active, and now evolution, so to speak, evolution got lucky, and so now we have an active, so we have a new protein that's a version of the original. And so again, everything's available online for download. We have a you can search, you can download. So we're making everything available, and there's a lot more in the manuscript. So this was the first big project that I wanted to talk about, but now I did want to switch, and I'm going to talk about this as a different project. It's a different type of work that we do, which is a more method development type of work, and maybe actually. So this is a tool we've developed to solve the metagenomics binning problem. So what's the metagenomics binning problem? So I'm going to illustrate with this cartoon. So this is the real world, or in a cartoon form, right? So bacterial genomes are these circular elements. They are, and bacteria live in these communities. And as you know, and here we can see, there's some, for example, the green ones seem to be very abundant, and the yellow ones seem to be more rare. And then some of them are intermediate. But then when we go in and we collect this data, what we get is these fragments, these short reads. And just to give you orders of magnitude, so the full chromosome is going to be the mega base pairs, and then the short reads is going to be hundreds of base pairs, so much smaller. Then we first assemble this into these so-called contigs, which are bigger chunks. And now we have maybe these chunks might now be in the thousands to tens of thousands of base pairs. So they're still much shorter than the full contigs. And for the most part, the assemblers don't make mistakes. They are, so here I've illustrated one contig as magenta and green. That's actually fairly rare that this happens. The bigger problem is that the genome is fragmented into all of these pieces. And so, and here in these cartoons, I'm showing you the colors, but in real life, the problem is we don't know what comes from where. And so that's the binning problem, is trying to find these groups where we try to put all of the contigs that originally come from the same chromosome or the same species together, while trying to not missing anything, and not getting contamination, right? And so this is basically, from the machine learning point of view, this is basically a clustering problem, right? We're trying to cluster things, right? We could also classify them. And that's also something that people do very regularly, where, and then from the machine learning point of view, we approach this as a classification problem. So we have some inputs, and we want to assign some classes. And so, and actually if classification worked, then this would be, we wouldn't need to do any of the binning. But there's two fundamental problems with classification. So one is, in a real sample, I might have a species that's just completely new, right? For my classifier, it's not going to ever be able to classify it because it doesn't even know that was a possibility, right? So if you have a species that's completely new, what would the classifier even be able to say, So here, and the other one is, again, as I said, humans, we are almost all identical. But if you look at, for example, E. coli, or you can have two E. coli strains, they're both E. coli, but they have about half of their genome is completely different. Not completely different genes, and so we might have, for example, here the magenta. Magenta might be a known species like E. coli, but there just might be completely blocks of E. coli genome that are completely unseen, never seen before. And this is actually really common. And so it depends on your data, but actually often the majority of contigs will not be classified, at least not at the species level, because of these two problems. There'll be species that we had never seen before, and there'll be variants that we had never seen before. And so Xiaojun, who actually now recently graduated, so he developed this idea of trying to merge these two approaches. So the classification approach on the one side, the binning approach, and so in doing something that's semi-unsupervised. And so the idea was to take on the one hand, try to do classification, and whenever we can be confident that two things we're very confident that they don't belong together, then we say they cannot be linked. And on the other hand, we artificially break up these contigs and to build. And if we have these two half contigs, then they must be linked because we are very confident that the contigs are parts of the same original chromosome. And so now we have this. We can have this as a learning problem, and we do this feature based, and we learn this embedding space where we try to embed our contigs such that things that must be linked are as close together as they can, and things that cannot be linked are as far apart as they can. And so we transform this original space, or actually we learn the transformation of the space, and then we apply the transformation of the space to all of the contigs, and then we perform clustering in this transformed embedding space. And the question is, does it work? And we compare this against other binning methods. And I'll be showing you several of these plots where we're comparing against other methods from the literature. And our tool is the green bars. And so if we have more green, means we have more higher quality genomes. And so if the green bar is bigger, that's good, basically. And so we have compared against so VAB was another sort of deep learning technique that had just come out, and MetaBat2 is maybe one of the most widely used methods. So the semi-bin really outperformed them and really gave really good results. The big problem with semi-bin 1, so this we published in 2022, and at the time it worked really well, gave really good results, but it cost a lot of computational, so it was slow. And part of the problem being slow is, or the bottleneck was that this initial classification of the contigs was one of the big bottlenecks. And so what we tried to do, something a little bit different, which was, let's just take pairs of contigs at random. And because unless we're unlucky, just most of the time, we are going to pick things that don't come from the same genome. So this has a couple of advantages. It's much, much faster. Also, there's no bias in terms of, it doesn't rely on prior knowledge, so we're not biased by the database. On the other hand, I mean, we're also not injecting any new knowledge, and sometimes we are going to be making mistakes. So basically, what gives? So when we have these sort of competing things, we have to look at the results empirically and ask, in the end, does it work or not? And so here now we're again looking at the green lines. So we're comparing against the previous version of our tool. And this is in some simulated data, and the green line, it's slightly better. So we do slightly better. And then when we look at real data, so this is the same, so again, we're using human gut, dog gut, so these two mammalian, ocean and soil as two environmental types. And across all of these, we do better. So the green bar is higher. And so we're much faster and slightly better. So this was really good. And the other thing we wanted to do is everything I've presented so far, all of the work was on short reads, so this Illumina data. But in the last couple of years, more and more people, including ourselves, have started to use this long read data. And frankly, you could always use semibin. It worked, and people did, but we hadn't really tested it. And so we started to look a little bit more into it. And at the end of the day, the biggest problem is that if you have short reads, you end up with much shorter context. So this is the same sample or 3 different samples that were sequenced with Illumina, with PacBio and nanopore, so Illumina being the short reads. And so with short reads, we end up with many contexts that are short. And with long reads, we end up with fewer contexts that are long, which means that we only need a much smaller number of contexts to cover a metagenome. And so our clustering was really not designed to give you small clusters. It expected, a lot of the parameters expected that the clusters would be large. And so what we did is it's maybe not the most elegant solution, but depending on whether you are using short reads, you get the same as before, this clustering approach, which uses an approach called InfoMap. But if you're using long reads, then we use dbScan, which can be tuned to give you very short or very small bins. So small in terms of number of elements. And again, we're going to be very empirical about this and ask, does it work? And so we looked at a lot of other different tools. Here, I'll just mention a little for fairness. Many of these tools were the authors don't say that they work for long reads. So it's a little bit unfair to even try them, but people were using them, so we tested them. And again, the green bar is higher, so semibin 2 does seem to give you more high quality bins than other tools, right? So the last thing I'll mention is that I'm here. I was presenting the methods, presenting some of the machine learning, but we do want to make this available as a tool, so even even people who maybe don't care anything about machine learning and neural networks, but just want to have very good bins, we've made it very easy for them. There's a lot of documentation. We have YouTube tutorials, and here, so here, if you follow the links, you'll see that I've written I've written a perspective on how we've maintained these over the long term, right? So with that, I'm also a little bit out of time, so let me just summarize. So we started by discussing. So we've looked at we tried to look at all of the genes from the global catalog at the same time. Most of them are rare and habitat specific, and as I mentioned, unfortunately, part of the genes that we find in multiple habitats tend to be things like antibiotic resistance. So more recently, we've been looking at the small proteins, again, with this perspective of looking at the global picture. And we focused on AMPs, antimicrobial peptides. But we were very successful with this machine learning model and at finding things that we validated in the lab, including in mouse models. And then I switched to a very different type of work where we've developed semibin, which is a machine learning system. And in semibin 1, we had this semi-unsupervised approach, and now we have a completely self-supervised approach with semibin 2. And if anything, we're all very, very empirical. For us, it really matters that we can be very thorough in evaluation, and we do produce really good results. So I just want to say, so a lot of this was done. So I showed you a lot of Celio and Ichan's work in the small protein. Xiaojun did semibin. Cesar and Marcelo were responsible for a lot of the MP work, but then all of the other collaborators. Funding, came from NSFC China when I was in China, and then since last year, the Australian Research Council funds my research. So thanks to them and thanks to you.
Thank you very much, Professor Lewis. This has been a great talk and a great overview about the global microgenome. I would encourage or ask members if they have any questions, you can just ask the questions or you can put them in the chat. or you can put them to me. Anything that you feel like. So in the next few minutes, we'll be asking Professor Lewis some questions. And I'll begin by putting forward mine. Prof, it looks like from the paper and from the presentation, the global microgenome is quite dynamic. Like when you look at the different map, the map where you collected the samples. So how do you ensure, since it looks it's dynamic from the research, how do you ensure uh, uh, the follow-up and making sure that all these different smORFs within different environments are mapped into the tool?
Yeah. So yeah. So yes, yes, it is very dynamic. Yeah. I mean, so one thing I'll say is we are using publicly available data, and out of that, maybe less than 1% is our own data, right? And so everything we know is biased by by what has been and so I showed you is biased by what has been sequenced. So this means, for example, that there's I mentioned the geographic bias, right? That there's less sequencing in some parts of the world than others. There's also the fact that we sequence a lot of human-associated samples and not other samples. So we are very species level biased, and so we are always sort of we only see what we've sequenced, but we probably have not, it's not an unbiased representation of the world, and it's nowhere near a complete representation of what the real world is in terms of what the biology is doing, right? So what we do is we keep updating this. So in this study we had 60,000 metagenomes. So in the Spire paper, which came out this year as well, but it's a slightly more up-to-date data set. It has 100,000 metagenomes. So we keep trying to update these things. We keep trying to update the resources, but we are playing a bit catch up with what's out there. And we only see what has been sequenced. I'm not sure if this answered your question, but I think I was trying to go in that direction.
Yeah, sure. There is a question here. How do you account for mutations in the identification of smORFs, especially considering the small proteins?
It's so yeah, so it's difficult. So we try to we try to collapse sequences if they are 90% identical. But 90% is a fairly high threshold. So this is actually something so with long sequences, this is a completely solved problem, right? There's many tools that do this and do this very well. We have a lot of understanding of how these things work. With short sequences, we just don't. I mean, even because again, so if you have two very long sequences, or not long, but like a couple of hundred amino acids, and they are amino acids, so your vocabulary is of size 20, and they are 50% identical over hundreds of amino acids. That does not happen by chance. That is very strong evidence that they have a common ancestor, right? Because you do not get 50% identity over hundreds of amino acids. But if your sequences are 50 amino acids, maybe it's by chance. You can get 25 amino acids by chance that are the same, and that is completely and now you can no longer, so the same sort of logic no longer applies. And so how to do these things, it's a big open problem. So this is something we're working on right now. We're thinking of, there's many different ideas, trying to look at not just sequence but structure, trying to look at not just the sequence of the protein itself but its neighbors, to give more information. But it's an open problem. How to do this? How to do these things? How to even for example, actually, I think that one I might have. Even for example, so I mentioned this in passing that we use a there it is. We use a machine learning model to try to find AMPs. So in the context of looking for function for long proteins, people generally don't use machine learning. They use sequence identity. Some sort of advanced forms of sequence identity. But this doesn't work for small sequences. And so there's a bit of context, but so here blue is BLAST, and then the others ones are 3 different machine learning models, including Macrel, which is our own. And you can see that so all of the machine learning models do better, much better than the BLAST. So again, we have had to go from looking at sequence identity to using machine learning as a solution for trying to assign function. So it's very much an open problem. It's something that
Thank you, Prof. As you presented, something came to my mind about protein structure prediction, but also function annotations. And we have huge models these days that predict protein structures like AlphaFold, but also combining that with function annotation of these smORFs. So is there a way that you can see AlphaFold being integrated into smORFs, but also coming up with these new smORFs, but also we can annotate their functions?
Again, some of that is exactly what we've been trying to do. So to be fair, function is maybe a little bit more limited here in terms of application, just because there's just you don't have as many amino acids to work with. So a lot of the structures are very simple, right? Because for example, often we see this, so you maybe have like a little helix and then a loop, and that's it. You don't have enough Lego pieces to build a big structure, right? Right. If you have a short sequence, it just doesn't. Nonetheless, it might still be useful to have some, the structure still matters. And it does seem that AlphaFold does work even for short sequences. So we'll be, I mean, when I say this is future work, we'll be looking at this over the next few years. So maybe ask me again in a couple of years. Maybe I can answer that question.
Okay, and I think that tags into the last question that was here. That even if small proteins they evolve quickly, how do you distinguish between the beneficial mutations that perhaps lead to functional changes and those that might not be or might be novel in sort of smORFs?
Yeah, I hate to say it again. That's an area of research right now. We're really trying to. So I can tell you that. So generally speaking, this area of research, we're not the only ones working on this. I mentioned Ami Bad's name, but there's a couple. It really has exploded in the last few years, trying to really understand small proteins. It was a little bit of an ignored field. Including our own work from in the microbiome from 10 years ago. We really didn't look at it at all, and then only about 5 years ago did we start thinking about it. Right now we don't know. And a lot of what we're trying to do is try to use the fact that now we have so much data, because again, so we do think that if you have a small open reading frame in one genome, that's probably not very meaningful. that might really not be that might not mean much, because again, it could really just be random. But if you have the same sequence conserved across many genomes and across metagenomes, and now we do have these very large data sets, now we can start to make statistical inferences about it. So trying to look for these cases is something that we're doing as well. But and the paper that just came out in Nature Communications, that was one initial, that's one initial, but this is ongoing work.
Okay. Prof, maybe lastly, and the good thing you're a computer scientist also from the background, for people who are passionate about like deep learning and then genomic data, what are the possible sort of machine learning extrapolations that you can see or you see in the future that can be done, particularly in this work?
So I think so on the, it's again some of what we'll be, I can tell you some of the stuff we'll be trying. So ideally we would have some sort of protein language model that we would be able to take a sequence and then will give us a prediction on function, or even better, we would give it a prediction on function and it would design a sequence for us. I think we're far from that, particularly in this regime of small proteins, because there's just not a lot of data. So some of it we will just need more data to solve it. What we're more hopeful over the short term is to try to have not a protein language model that looks just at the protein, but looks at the protein and its surrounding context. So that so in bacteria, proteins are generally not they're grouped together in the genome in some sort of logical, so that things that are functionally related end up next to each other, at least statistically. So this is not. So some people exaggerate how conserved this is, but statistically they will tend to be closer to each other. And so what we're thinking now is can we have models that exploit this? So they're not really. So ideally, it would even be some sort of protein language model that's not just an individual protein, but sort of also protein and gene neighborhood model. So this is what we're thinking nowadays. But at the same time, we're also trying to just get more data. I think some of these problems we just don't have a lot of data.
Thank you very much, Prof. This has been great and fantastic, and a great beginning for all of us. So we have guys here in statistics and bioinformatics, PhD student, and medics. So the sky's the limit. Thank you very much.
Thank you. I mean, and as I said, I've left the link, but if there's anything, anyone feel free to email me, and we can even set up another Zoom one on whatever. I'm very happy to get questions, and I'm very happy to have been invited. Especially, I didn't even know it was the first one, so I'm very honored for that. Thank you.
It's the first, and your name will be placed in our group somewhere.
Thank you.
All right, guys. Prof is heading. It's night time, I guess. So I should say good night.
It's night
in Australia. Yes. All right. Have a great night, and bye-bye to everyone here.
Thank you.
Bye-bye.